proteins. I've already commented to you that most organisms, including us, are 70% water. So if you take out the water, that's dry weight, what's left over is dry weight. 70% of our dry weight is protein. So take out the water, 70% is protein. For us, muscle enzymes are always protein. Again, parts of cell membranes are protein, and so on. But enzymes and proteins, enzymes and muscle alone, that's quite a bit of protein. Proteins are made up of amino acids. Those are the subunits. And amino acids look like this. Over here, carboxyl group. That's why it's an acid. Carboxyl groups are acidic. That made the fatty acid an acid, it's making the amino acid an acid, the carboxyl group. In the middle here, we have a carbon with an R on it. I'm sure many of you are trying to figure out what's an R. You're trying to think of the periodic table. Stop thinking about it. It's not on the periodic table. We'll come back to that. Here is another chemical group. As you're trying to figure out, there's a lot of chemical groups. And a very well written N too. Thank you. We like no judgment. NH2 is an amino group. The AMIN means nitrogen. So what we have is a carboxyl group on one side, an amino group on the other side, and in the middle, carbon with an R group. We'll talk about the R in a minute.
Quiet.
Now, it turns out that with all the life forms on Earth, there are only 20 amino acids. In all of nature, there are 20 amino acids. And they come together in different combinations, just like letters making up a word. And again, only 20 make up all the proteins on Earth. It turns out that even more remarkable than that, what I've just circled is identical in every amino acid on Earth. Amino acids are identical, except for the R. So every amino acid has a carboxyl, has the amino, has that carbon in the middle. So the obvious question is, what is the R? Well, I just said there are 20 amino acids, and every amino acid is identical except for the R. So the R is what determines the amino acid. The R is what determines the identity of the amino acid. If you change the R, you change the amino acid. be some examples of R's, just so you kind of know what they are. This is off the record, so it's just for information, just so you kind of have an idea. The R could be, I said, still off the record here. So each carbon, and then some R group.
What is this amino acid called?
Pardon me?
R. What is this one called? Or is it just?
It's just called an R group. That is what it's called. But it is basically, well, it's not an unknown because they know what they are. But depending on which one you got, you have a different amino acid. Yeah, it's just called an R group.
Do we have to know all of them?
You have to know any of them. That's why I said it's off the record.
So
You don't have
like
to know
there's a
any.
page in here, all of the 20 in here.
I know. That's why it's off the record. You don't have to know any of them. You know, to me, that's reference book material. If you ever need to know the R group, go look it up. I don't know what most of them are either. I don't care. So, but just know that it's there. Know that the R is there. That's your amino acids. No, they don't. They don't.
So amino acids are chains of proteins?
Amino acids make up proteins.
So
We're
amino-
gonna do that right, huh?
Yeah, amino acids make up the proteins, and depending on what kind of protein it is, so what kind of, just like for example, like what, like would it be like a chicken or something, or like is that like a protein, or like what
Well,
would be?
DNA
Oh.
would
Oh, so it's
determine what protein you get. So the protein would make up your chicken or whatever,
yeah,
but something has to give the instructions of what protein.
that's what that does.
So the DNA does that. DNA determines proteins. But the protein is just simply what you're coming out with. Anyhow, proteins are made up of amino acids. So all you do is you add one amino acid after another. It's like adding cars to a train. So we need another amino acid. By this time, you're kind of accustomed to doing condensation reactions. We're going to do it again. But for the first time, this one's a little bit different. Because we've always had two hydroxyls before. If you think about it, we always did a hydroxyl on one side and hydrogen only on the other side. Well, we can still do that. See, that's still there. The only thing that's different is that always before we had an oxygen left over. This time we do not. But that's okay. It's not required. So take out water like you've always been doing. What's left over is nitrogen, not oxygen, but that's okay. And then connect it. So actually, you do the same thing you've already been doing. Yeah. I'm sorry, what? So dipeptide simply is two amino acids. The bond connecting the two amino acids is called a peptide bond. So that's a peptide bond.
Inside, so that bond is called the dipeptide.
No, the
No, that bond
is
is
called
called
a
a
peptide
peptide
bond. Peptide bond. But the entire thing is a dipeptide. You have two amino acids.
You have taken like two H's from the NH3.
There is no NH3.
I mean NH2. Have you taken those two hydrogens
Over here.
and the oxygen?
Sure.
When you combine.
Well, yeah, because there'll be another amino acid over here. You just keep doing it over and over again.
Right, I guess what I'm asking, does it matter if you take one hydrogen from each? Or could you
Well, you have to take a hydroxyl from one.
you have to take both of
You
each?
gotta take water.
Okay.
You Gotta be water, H2O. So,
Oh.
to answer your question, no, you gotta take OH from one, H from the other.
Okay, so you could take H2 from one
Now I see where you're going. No.
Yeah, that's.
Hydroxyl from one, H from the other. And like I was saying, then you just keep doing it. You can have a tripeptide, tetrapeptide, et cetera, eventually polypeptides, so many peptides, and eventually a protein. You keep adding on amino acids, sooner or later you get a protein, and there's an obvious question. How many amino acids do you need for a protein? There's an obvious answer. I don't know. I don't know. Once again, it varies. Well, you've been hearing that from me a lot. It varies. The smallest protein I know of is insulin. 51 amino acids. It's the smallest one I know of. The next smallest is hemoglobin. HB, hemoglobin. 143.
That's just what you know of, so are you saying that there are smaller amino acids?
I really don't think so.
Okay.
I really don't think so.
So peptide just means that group right there.
Yeah. Yeah. So like I say, you keep adding on amino acids or peptide. Sooner or later, you've got your protein, but that can vary wildly. If these are the two smallest ones, what that tells you is proteins are hundreds, if not thousands, of amino acids long. And many proteins are thousands long. They're huge. Now, for example, let's say we did have a protein, 1,000 amino acids. And let's say we took out one. Now it's 999. This is no longer a protein. It's incomplete. This is now polypeptide. Correct. A polypeptide is an incomplete protein. I'll try to repeat the question, actually. It's kind of hard for me to do that. Yeah, she was just asking the polypeptide that is no longer a protein. And the answer is yes, it's no longer a protein.
So it's not the case that 1,000, going from 1,000 amino acids to 999 amino acids doesn't make it another type of protein?
Well, it might. Now, his question is, could we maybe be making another protein? It's possible, conceivable. It's not hugely likely, but anything's possible. But generally not. But the problem is, even if the sequence is identical between a protein of 999 and a protein of 1,000, maybe the bending is not the same. They have different shapes. So it's conceivable, but it's not hugely likely. Now, by taking off one amino acid, you're still be making another protein. I would say probably not. But not 100%. It's possible. So at any rate, polypeptide is an incomplete protein. Incomplete.
Can you repeat that? It's an incomplete what?
Yeah,
Protein,
just
okay.
polypeptide is an incomplete protein.
1,000 is amino acid, full amino acid.
That would be 1,000 amino acid is my example of a full protein.
Okay.
So if you take one off, it's no
It's
longer full. It's incomplete.
Is there any way to look at an amino acid chain and know from just looking at it?
Of amino acids, just like trains are made up of cars. Well, it's true, it is not complete. What I've actually given you is called primary structure. primary structure is a linear sequence of amino acids. In other words, a straight line of amino acids. You ever stop making those on dead meat? Linear sequence of amino acids, like a train, but that's not the case. I mean, yes, it's true, but it's not just a straight line. We also have secondary structures. So we still have the sequence of amino acids. You'll always have it. The secondary structure can be either a helix, like a spiral staircase, or it's called a pleated sheet. A pleated sheet. I've actually never seen a pleated sheet, but I guess it looks like that if you never saw one. So those are secondary structures. Usually a helix is the most common, but a pleated sheet is possible. So you still got your amino acids, it's just not in a straight line. Spiral. But you know, it's still kind of linear. It's still kind of going in one direction. And proteins don't do that. So we have tertiary structure. Tertiary structure, we still have our sequence of amino acids, still have our helix or whatever, but it's in a three-dimensional shape. You got your helix, but it's not just straight up and down. It's, pardon me? Three-dimensional shape. I thought I wrote that, I did not. 3D. So it gives the molecule shape and structure. The helix is all twisted and bent, convoluted, twisted. That's giving you a three-dimensional shape. The shape of a molecule is a big deal. You're going to run into that again. The shape of a molecule helps determine the function of a molecule. The shape of a molecule helps determine the function. If you change the shape, you change the function. Probably, you deactivate it completely. So again, if you change the shape, you are going to change the function, probably deactivate. Now, easy example, putting your key into the lock. Well, obviously, the key fitting into the lock is based upon the shape of the key. If you melt down the key, it's not going to fit, which means it no longer works. So many molecules work just like that. Enzymes in particular, proteins, are based upon shape. If the enzyme is the wrong shape, it doesn't work. Now, in fact, in making an enzyme the wrong shape, this is called denaturation. Denaturation. Denaturing a protein is changing the shape of the protein. So if you denature a protein, you're changing the function, and again, probably deactivating. Maybe not, it's not always guaranteed, but there's a really good chance of it.
So like when you burn, like, the steak, is that denaturing burning the steak?
That would be denatured, and you're not gonna renature it, yes. That would be definitely denaturing your steak. In fact, that brings up a point. How do you actually denature a protein? I'm kind of making it sound easy, but how do you actually do it? Actually, it is easy. And he referred to one easy way, temperature. And I'll bet already many times in other classes, you have done labs, you were heating something, you were probably denaturing a protein. So temperature will denature a protein. Acid, change the pH. Salt. As you can see, these are easy. It takes 2 seconds. Also, alcohol can denature proteins. Now, I don't mean drinking alcohol. If that were the case, I wouldn't have a class. But what I mean by this is, like when you go to the doctor, you're going to get a shot. So he rubs your arm with alcohol first, and then he sticks you. The alcohol will denature proteins in bacteria. So it kind of sterilizes your arm a little bit. So alcohol can denature. So anyhow, very, very simple. And I'm just commenting to renature. Not so simple. So for example, taking something off the heat. Will the protein go back to the way it was? No. Usually not. Now sometimes yes, but usually no. If you take off whatever, probably whatever has occurred, you're stuck with it. Easy example, cooking an egg. You break the egg, you've got the yolk and the clear stuff around it. Once you cook the egg, it turns white. And then you take it off the heat, it's still white. Well, the white is your denatured protein. And taking it off the heat, it's still denatured. So there are some proteins that will renature. It's not rare, it's not common. It's pretty rare. I'm sorry. There are proteins called allosteric proteins that will do it. That chemotactic protein, yeah, so
Yeah, I know exactly.
that's why I wasn't giving an example. At any rate, so this is how you do it. You might be curious, why would you want to do it? Why would you denature? Actually, you guys, as healthcare people, maybe taking care of patients, probably you will not do this. Denaturing your patient's enzymes is not your best path to advancement. So probably you will not do this. But in the research lab, we did it all the time. When I was still doing my research, sometimes I'd be working on something, looking at some reaction, and the reaction would be going so quickly that I really couldn't get my results. So I had to slow things down. One way of slowing things down would be to denature the enzymes, slows down the reaction, then I would get better results. So research labs can do that. You guys, again, as healthcare people, probably not. But at least be aware that it's there. Yeah.
So when you're cooking eggs, you denature.
That's denaturing. I'm sorry. Have primary, secondary, tertiary structure. They've all got a sequence of amino acids. They've all got that helix or pleated sheet, and they all have the tertiary structure. So all proteins have primary, secondary, tertiary. Now, maybe, sometimes, but not very often, you have quaternary structure. This is not common. Quaternary structure is when the protein has subunits, and the subunits are proteins. So again, the protein has subunits, but the subunits themselves are proteins. So in this case, each of these is a protein. So each of these has primary, secondary, tertiary structure. But all together, they make up a bigger protein, the quaternary protein. Best example is hemoglobin. Hemoglobin does this. So each of these is a protein by itself. But all four of these together make up hemoglobin. Actually, as far as I know, bacteria do not have quaternary proteins. We do. So again, all proteins, primary, secondary, tertiary, they all do. Quaternary is a maybe, and frankly, it's a not very often.
Say it one more time.
I don't know what I said.
You said the
Bacteria don't.
Oh, yeah, bacteria
Bacteria.
don't really have quaternary proteins, but we do, and hemoglobin is the prime example. But it's not a very common structure.
and the quaternary is
When you have
is
subunits,
a subunit of a protein of a.
but these, the subunits are proteins. So each of these is a protein. And then the four proteins together make up a bigger protein. Wait, hemoglobin in eggs? No. Albumin, I said. Albumin is the egg protein. Yes, yes. So I'm sorry, go ahead. All proteins are made up of amino acids. The quaternary proteins are made up of smaller proteins. So each of these is a protein. By itself, each one is simply a protein. But all together, we would have hemoglobin or some other quaternary molecule. So quaternary proteins are made up of smaller proteins. Renaturation is very rare. I'm just simply saying, I'm not saying that it never occurs, because it can. But usually no. Well, that was my example of the egg protein. Once you denature, it's done. So, possible sometimes, but not common. It happens because some proteins are made to renature. And we're actually going to talk about that at a later time. They're called allosteric, but they're made to renature. But mostly they don't. Mostly. Okay, so be prepared to draw a dipeptide. So now you've got three things you've got to be ready for. To draw a disaccharide. I'll give you the monosaccharide. Be prepared to draw a triglyceride. And be prepared. Sorry. And be prepared to draw a dipeptide. One of those three, there's just an extraordinarily high chance that one of those will be on your midterm.
What is it?